Hello everyone, welcome back to Sedu Satsavi YouTube channel. So today we are launching a new MCQ series for the Food Safety Officer exam called as FSO exam, which has been recently notified in the Kerala PSC exam. And this series is called as FSO 1000 MCQ series, where we'll have 10 MCQ discussion in each video. That means by 100 videos, we will be able to cover 1000 MCQ series. So that is FSO 1000 MCQ series. So this will include Q&A discussion from all the subjects given in this syllabus. There will be easy tips and tricks to remember the concept, and there will be effective discussion to make the concepts clear. So today we'll kickstart with a small portion of microbiology subjects, and without any further delay, let's move on to the first question of the day. So the question is, which of the following method removes pathogens including spores? A. Disinfection. B. Sterilization. C. Both A and B. D. Germicide. So you need to remember both the disinfection as well as sterilization can remove pathogens. So disinfection will remove pathogens. Sterilization will also remove pathogen. Then what is the difference between disinfection and sterilization? Sterilization, apart from removing pathogen, can remove the spores also. It can remove spores. Whereas disinfection do not remove spores. Disinfection means it do not remove the spores. Whereas sterilization can remove spores apart from removal of pathogens. So there is a difference between disinfection and sterilization. So what would be the correct answer for this question? Yes, definitely B choice. And what about germicide? Germicide. Side means kill. Okay. Killing of germs is called as germicides. Okay. Now let's move to the second question. Which of the following is not an example of physical method of sterilization? A choice, gamma rays. B, tyndallization. C, glass filtration. D, glutaraldehyde. E, all of the above. So the first thing you need to know the different sterilization method. So basically the sterilization can be divided, or the method of sterilization can be divided into two categories. One is the physical sterilization. The second one is called as the chemical sterilization. Now let's look into what is physical steril or the physical method. What are the types of physical method of sterilization? So there are of three types mainly. One is the heat sterilization. Second physical method is the filtration kind of sterilization. The third one is the radiation sterilization. Now, in this heat sterilization can be divided into two categories, dry heat and moist heat sterilization. Dry heat sterilization and moist heat sterilization is there. Now, when it comes to dry heat, the examples are gas flame. Gas flame is a dry heat sterilization. Sunlight, in fact, is considered to be a dry heat sterilization. Hot air oven. Hot air oven is an example for dry heat sterilization. Incinerator is also an example for dry heat sterilization. Whereas when you come to moist heat sterilization, basically we can divide it into three categories based on the temperature. So one method is less than 100 degrees Celsius. The second one is at 100 degrees Celsius. And the third one is more than 100 degrees Celsius. So at these three different temperature, we can do a moist heat sterilization where there will be moisture will be there, latent heat would be there. So that is the difference between dry heat and moist heat. We'll talk in detail in the coming slides. Now filtration sterilization can be divided into depth filters, depth filters. Another example is membrane filters, also the air filters. So these are the examples for filtration sterilization. Now coming to the radiation sterilization, that can be again divided into two categories. One is the ionizing radiation. The second one is called as non-ionizing radiation. Now in the ionizing radiations include the ionizing radiation includes X-rays, gamma rays, and the cosmic rays. Whereas the non-ionizing radiations include the UV as well as the infrared. So these radiation can also be used for sterilization, and these are the various methods. Okay, now we'll go to the chemical method of sterilization. So far we have seen the physical method. So in chemical method, we use chemicals like we can use alcohols, we can use aldehydes like formaldehyde, molecular formula is HCHO. We can use glutaraldehyde. Also, the phenol can also be used as a chemical method of sterilization. We can use halogens, we can use oxidizing agent. In gas sterilization, where we use ethylene oxide, it is also a chemical. So it is a chemical method of sterilization. We can also use dyes. So that is so what I have told is an overview of physical method of sterilization and the chemical method of sterilization. Now let's look into the question, which is the following is not an example. So this gamma rays belongs to a physical method of sterilization. Tintilization is actually a physical method of sterilization. Glass filtration is also a type of physical method of sterilization. Whereas you look at glutaraldehyde, we already told it's a chemical, right? Aldehyde. So the correct answer for this question will be glutaraldehyde. Glutaraldehyde is not a physical. In fact, it is a chemical method of sterilization. Now let's move on to the next question. The third question, the preferred temperature and duration in sterilization using hot air oven. A choice 160 degrees Celsius, B choice 170 degrees Celsius for 1 hour, C choice 180 degrees Celsius for 0.5 hour, D all of the above. Okay. Now the first thing you need to know about hot air oven. His hot air oven is a dry heat sterilization. Okay, we told that we need to understand that hot air oven is a dry heat sterilization. Okay, so the mechanism involved in using hot air oven sterilization, you can remember the mnemonic doc. First, the D stands for denaturation of protein, O stands for oxidative damage, C stands for charring. So this is the mechanism by which how it sterilizes the products. Okay, so you can remember the DOC, denaturation, oxidative damage, and charring. Now coming to the temperature and duration, the most commonly employed temperature is 160 degrees Celsius for 2 hours. This is the common thing which is used in a hot air oven for sterilization. However, if you can increase the temperature from 160 to 170 degrees Celsius, what we can do is that we can reduce the duration. That means 170 degrees Celsius for 1 hour can also be used in hot air oven. Also, you can use a still higher temperature, 180 degrees Celsius, then the duration could be reduced to zero 0.5 hours. That means 30 minutes. So these three temperatures and duration can be used in hot air oven for sterilization. And what is the use of this hot air oven sterilization? Normally, it is used to make used for the sterilization of glasswares, wax, oils, powders, gels, dusting powders, paraffin wax, metallic instruments, cotton swabs. So it is used for sterilizing these substances like glasswares, wax, oils, etc. Okay. Now we'll move to the question, the preferred temperature of duration. So I told you the normal temperature and duration is 162 hour. Also, if you increase the temperature, you can reduce the time. So all these are correct. So the correct answer for this question is all of the above. Okay. Now let's move on to the next question. Human anatomical waste are sterilized by A. Hot air oven, B. Choice incineration, C. Choice inspissation, D. Choice kindlization. So remember, all the hospital waste are sterilized using a method called as incineration, which is a dry heat sterilization method. So incineration is a dry heat sterilization method, and which is used for used for the sterilization of all hospital waste, in also the human waste, all human waste, animal waste, soiled beddings and dressings, expired anti-cancer drugs or cytotoxic drugs. All these are sterilized using this incineration method. Okay. Now, what is this incineration? So I told you it's an example for dry heat sterilization. It is an example for dry heat sterilization where you will pass the substance through two chambers. So in the first chamber, the temperature will be 700 to 800 degrees Celsius. From this first chamber, it will be transferred to another chamber where the temperature will be more, that is 1000 degrees Celsius to 1100 degrees Celsius. Okay, so this is how incineration is done. Okay, two chambers and at a very higher temperature like 700 to 800, and the next chamber they will use still higher temperature 1000 to 1100. Okay, now let's move on to the question. Human anatomical waste are sterilized by, as I told you, all the human anatomical waste, animal waste, hospital things are all sterilized by incineration method. So the correct answer is B choice. Okay. Now let's move on to the next question. That is the fifth question. Which of the following is an example for moist heat sterilization? A. Autoclaving. B, tindalization, C, inspissation, then D, all of the above. Okay, so the first thing you need to know, this moist, I told you the heat sterilization can be of two types, dry heat and moist heat. Dry heat, we have already told, the hot air oven and incineration. Now coming to the moist heat sterilization, I told you based on the temperature, we can divide into three categories: less than 100 degrees Celsius, at the 100 degrees Celsius, at the 100 degrees Celsius, and which is also you can use a higher temperature, that is more than 100 degrees Celsius. Now at less than 100 degrees Celsius, you have three different things. One is inspissation, the second one is pasteurization, the third one is called as vaccine bath. So this comes under less than 100 degrees Celsius. Now at 100 degrees Celsius, you have two things. One is boiling, that is at 100 degrees Celsius. Also tintalization, that is also done at 100 degrees Celsius. Now at more than 100 degree, autoclaving is the moist heat sterilization method used. Now let's come to the less than 100 degree. So I told you inspissation, pasteurization, vaccine bath, they are done at less than 100 degrees Celsius. Now inspissation, you can remember the 80, 20, again 80, 20, 80, 20. Okay, so what does it mean? 80 degrees Celsius for 20 minutes, that is the first day. Again 80 degrees Celsius for 20 minutes, that is on the second day. Again, the 80 degrees Celsius for 20 minutes on the third day. So that means three consecutive days, you need to sterilize it using this 80 degrees Celsius and 20 minutes. And that is called as inspissation. Okay, so that is inspissation. Now coming to pasteurization. Pasteurization also, it is a example for moist heat sterilization at less than 100 degrees Celsius. So normally pasteurization is done for milk. You all know that it is done for milk. Now, what is the temperature and duration? So normally it can be done at a 63 degrees Celsius for 30 minutes, and this method is called as Holder method. Okay. So pasteurization using Holder method, the temperature is 63 degrees Celsius, 30 minutes. Or you can also increase the temperature to 72 degrees Celsius and for 20 seconds and immediately cool it to less than 13 degrees Celsius. So that is also another way to do pasteurization. 72 degrees Celsius for 20 seconds, immediately cool it to a less than 13 degrees Celsius, and this method is called as flash method. Okay, so two ways normally we can do pasteurization: holder method and flash method. And which is the better method? Yes, flash method is a better method compared to holders method. Okay, just need to remember the temperatures in both the methods. Okay, now vaccine bath, which is normally carried out for bacterial vaccine to make it heat inactivated. Okay, so that is the purpose of vaccine bath, to make it inactivate the bacterial vaccines, to you can make it heat inactivated, and the temperature used is 60 degrees Celsius for 60 minutes. Okay, so that is what is done in the vaccine bath. So all these inspissation, pasteurization, and vaccine bath is example for moist heat sterilization less than 100 degree Celsius. Okay, now there are moist heat sterilization which is done at 100 degrees Celsius, right? So which are the examples? Boiling and tintalization. Now in boiling, the temperature is 100 degrees Celsius, 15 minutes. Whereas tintalization, you need to do 100 degrees Celsius, 20 minutes in the first day, again 100 degrees Celsius, 20 minutes, second day, again 100 degrees Celsius, 20 minutes for the third day. So that means 3 days you need to do tintalization, 120, 120, 120. Okay, what is the difference between boiling and tintalization? In boiling, you just do only for one day, that is 100 degrees Celsius for 15 minutes. Therefore, this will not, it do not kill spores. It do not kill spores. Whereas tintalization, since you are doing it for 3 days, 120, 120, 120, tintalization can kill the spores. Tintalization can kill the spores. Okay, so this is the moist heat sterilization which is done at 100 degrees Celsius. And which is the moist heat sterilization which is done at more than 100 degrees Celsius? Yes, the answer is autoclaving. In autoclave, normally the temperature used is 121 degrees Celsius for 15 minutes and 15 psi pressure. Okay, so remember 121, 15, 15, 121 degrees Celsius, 15 minutes and the 15 psi pressure. But also remember, if you wanted to kill the prions, which is very much usually resistant, okay, if you wanted to kill prions using autoclave, then the temperature should be 134 degrees Celsius and the time should be 1.5 hours. Okay, so to kill the prions, the autoclave temperature is 134 degrees Celsius or 1.5 hour. Normally 121 degrees Celsius, 15 minutes and 15 psi. Now autoclave is normally used to for the sterilization of aprons, non-sharp instruments, sputum, and mostly the sutures. All sutures are sterilized using autoclave method. Okay, now let's come back to the question. Which of the following is an example for moist heat sterilization? So autoclaving is an example for moist heat sterilization. Tintilization is an example for moist heat sterilization. Inspissation. So the correct answer for this question will be D choice. Okay, now let's move on to the sixth question of the day. Which among the following is a wrong combination of temperature? A. Pasteurization Holder method, 72° 20 seconds. Tintilization 100 degrees Celsius 20 minutes. Autoclaving 120 115 minutes. D. Inspissation 80° 20. So I told you inspissation 80 20 80 20. So that means this combination is correct. Autoclaving I told you 121 degree 15 minutes 15 psi. That means it is correct. Tintilization I told you 120 120 120. So that is also correct. So now look at the A choice. Holder method I told you. Holder method is normally done for 60, done at 63° Celsius for 30 minutes, and 72° Celsius 20 seconds is called as flash method. So this is a wrong combination. So the correct answer for this question is A choice. Okay, whereas B, C, D are the correct combination. Okay, now let's move on to the next question. Which of the following tests check the efficacy of pasteurization? A. Phosphatase test. B. Coliform test. C. Both A and B. D. LJ media test. Now you know that this pasteurization is done for milk, for the sterilization of milk. But how would you understand that if the milk is sterilized properly? Okay, so we have two tests to check the efficacy of pasteurization. Two tests. One is called as the phosphatase test. Okay, so phosphatase is an enzyme present in the milk. Now, once it is sterilized, this enzyme will not work. So, if you add any substrate, that will not change the color. So, that test is called as phosphatase test. Okay. Now, the second method to check the efficacy of pasteurization is called as coliform test. So, where in coliform test, you can check for the presence of E. coli, also the Klebsiella. Okay, so these are the two tests commonly used to check the efficacy of pasteurization: phosphatase test and coliform test. So the correct answer for this question will be both A and B. Now let's move on to the eighth question. Which of the following is an example for filtration sterilization? So I told you physical sterilization can be heat sterilization, filtration sterilization, and radiation sterilization. Now filtration sterilization, basically we can you can remember this mnemonic MAD, MAD. M stands for membrane filter, A stands for the air filter, D stands for depth filters. Now, in membrane filter, normally the reagents or the chemical what is used is cellulose acetate or cellulose nitrate. Okay, so this is the filter what is used in membrane filter. So the membrane filter is made up of cellulose acetate or cellulose nitrate. Now, coming to the air filter, you know that HEPA. What is HEPA? HEPA is nothing but high efficacy particulate air filter. High efficacy particulate air filter is called as HEPA. So HEPA filters are used to is a part of filtration sterilization. Now the efficacy of HEPA is 99.97%. That is the efficacy of HEPA. Now what is ULPA? ULPA is ultra low particulate air filter. Okay, ULP is ultra low particulate air filter. The efficacy is more than HEPA. It is 99.99%. Okay, ULPA and HEPA, they are air filters. Okay, now coming to depth filters, the examples are candle filter, seeds filter, and glass filter. Nowadays, the seeds filters are not used nowadays. Not used. Why? Because it is made up of asbestos, a chemical called as asbestos, and you know that asbestos is carcinogenic. They can cause cancer. So that is why seeds filters are not used nowadays. Normally, this heat filtration sterilizations are normally used to for heat to make to sterile this heat sensitive material. So if this compound is heat sensitive and you need to make it sterile, you need to better to do a filtration sterilization. So normally the vaccines, antibiotics, serum, toxins are sterilized using filtration sterilization. Membrane filter or depth filters are used. Okay. So the coming to the question, which of the following is an example for filtration? So seeds filter is an example, glass filter is an example, membrane filter is an example. So the correct choice is all of the above. Okay. Now let's move to the ninth question. Cold sterilization is done by A. IR radiation, B. UV radiation, C. Ionizing radiation, D. All of the above. So first thing you need to remember is cold sterilization belongs to radiation sterilization. Radiation sterilization. So basically I told you heat sterilization is there, filtration sterilization is there, radiation sterilization is there. That comes under the physical method. Okay. Now this in radiation sterilization, we can divide it into two categories. One is the ionizing radiation and the second one is the non-ionizing radiation. And which are the examples for ionizing radiation? X-ray is an ionizing radiation, gamma rays, cosmic rays. Whereas non-ionizing, it is UV and infrared rays. Now, the ionizing radiation have high penetrating power, whereas the non-ionizing radiation have a low penetrating power. Now, since this ionizing radiation have high penetrating power, they can kill spores. They are sporocidal. Ionizing radiations are sporocidal, whereas the non-ionizing radiations are non-sporocidal. They cannot kill the spores. Now, one thing you need to know, this ionizing radiation do not increase the temperature of surface. So since it do not increase the temperature of surface during sterilization, it can be called as a cold radiation. Now, if you look at the non-ionizing radiation like UV or IR, it can increase the temperature of surface during sterilization. It can increase the temperature of surface. Therefore, since it increases the temperature, it is considered to be a hot radiation. Okay, so these are the difference between ionizing radiation and non-ionizing radiation. You just need to remember the difference and examples. Okay, now normally this radiation sterilization is used in the OT, operation theater fumigation, biosafety cabinets, and all that. Okay, now let's come to the question cold sterilization. So I told you cold sterilization is an example for ionizing radiation. So the correct answer for this question is C. Coming to the last question, the control used for filtration sterilization is A. Brevendi Brevandimonas diminuta, B. Bacillus subtilis, Bacillus stereothermophilus, D. Bacillus mimilis. So you need to know we always have a control to check whether sterilization is done properly and all that. To have a control, we have biological controls for each sterilization. So in the case of hot air oven, we can use either the biological control like Bacillus subtilis or Bacillus atrophius or Clostridium tetani can be used as a biological control. Now coming to autoclave, you can autoclave moist sterilization. The biological control is Bacillus stereothermophilus. Okay. Now, in the case of filtration sterilization, it is Brevandimonas diminuta. That is the biological control used in the case of filtration sterilization. Now, coming to radiation sterilization, the biological control is Bacillus humilis. Okay, so here the question is, which is the biological control for filtration sterilization? The correct answer is A choice. Okay. So, hope you understood this 10 MCQ discussion. Keep on watching. Thank you.